Hello everyone, welcome to my YouTube channel. This is Enoch's Tutorials. In today's video, we are going to look at the methods or the technology called the recombinant DNA technology, which is more of a concept being basically studied under molecular biology and genetic engineering. So don't forget to like, subscribe, and also share the link after watching so you can receive every kind of update from this channel. So let's look at first the recombinant DNA technology, the meaning. We will look at the tools involved in the technology and the procedure that is involved in the method. So what is recombinant DNA technology? So as the name implies, we say it is recombinant. So the word recombinant means that we move of recombine two DNA from different sources. So here we define it as a genetic engineering procedure that is used to produce an artificial DNA through combination with different genetic materials. So you can have two different DNAs and we recombine them to form one DNA. Now the newly formed DNA, which is from the two, is called a recombinant DNA. So the recombinant DNA technology, we say it is a method that is used to produce artificial DNA through combination of different genetic materials or different DNAs. So let's say we have this DNA strand, this one, from let's say a human cell, and we recombine this human cell or human DNA with let's say a DNA from a bacteria. We form this one as the recombinant DNA. We say it's the recombinant because it's made of two DNA sources, one from bacteria and one from human source. So basically, this method is used to amplify a particular DNA strand in the body of a bacteria. So here we insert this DNA strand in one other strand as a plasmid, and you insert this plasmid into a bacteria, and upon division or the binary fission of this bacteria produce a copies of this bacteria, which will all we believe will contain this our gene of interest. So this method is basically used to produce a large copies of our gene of interest in vivo, that is inside a cell. So as seen here, this one is a plasmid from a bacteria. This section is where we insert the bacterial DNA or the human cell DNA. Then we produce a recombinant DNA. So this one is the recombinant DNA because it is made of DNA strands from different sources. If you want to produce a particular protein which is let's say deficient in one organism, and you are able to search for the genetic code or let's say the gene that codes for that protein, you can amplify that gene in the body of a bacteria, and that bacteria can be used or can use that gene to synthesize protein. Let's say we have insulin and you're able to identify the gene or the gene that codes for insulin synthesis, and you incorporate this insulin gene in the body of a bacteria plasmid, and you insert this plasmid with a recombinant DNA in a bacteria. This ones will have this gene, and they can use this gene to synthesize chromosomes, and these chromosomes can be used to synthesize protein, which is the insulin, in the body of the bacteria, which you can get a lot of the insulins to be isolated and used to basically cure diabetes. Now let's look at the tools that is involved in recombinant DNA technology. So if you want to recombine two DNA strands from different sources, you need some tools before the procedure can work out. And these tools are what we say the tools required for recombinant DNA technology. The first one is called a registration enzyme. We have the polymerase and ligase. We have the vectors and also host organism. Let's take them one by one and look at what they are. The first tool for the recombinant DNA technology is what they call the restriction enzymes. Now these enzymes are called molecular scissors, and they used to cut sections of DNA strands. So they have specific sites on DNAs which they are used to cut that section. So the word restriction enzymes, they are molecular scissors or enzymes that are used to restrict or cut some segment of the DNA strand. So here you can have this as a DNA strand. This portion can be cut using a registration enzymes. We have two types of that. We have the one we call endonucleases, the one that cuts inside the DNA strand, inside like this. The word endo means that it is inside. So endo means it cuts inside the DNA strand. And you can have an exonuclease, which is cut outside. So this one is cut at the end of the DNA. So exonucleases like can cut a segment of DNA strand like here, the end here, whereas the endo one will cut the inside of the DNA strand. A known example of the restriction enzyme, which is mostly used in molecular biology, is the E. coli R1. This restriction enzyme is basically found in E. coli and is the most used restriction enzyme in molecular biology. Now, this restriction enzymes basically cut in two phases. It can cut to give what we call a blunt end, or it can cut to give a sticky end. This type of cutting, as shown here, where this section is more like a slanted, also just like this. If each end of the cut is slanted like this, then this end is called a sticky end. A sticky end. In other words, if the cut is just straight without any slant, we call it a blunt end. And different restriction enzymes cut in different ways. The E. coli R1 is the most used restriction enzyme, which mostly cuts with a sticky end cutting pattern. So the restriction enzyme is basically the type of or the enzyme or the molecular disease which is used to cut the segment of the DNA and insert another DNA strand inside there. The next one is what we call the polymerase and the ligase. So the polymerase is the enzyme which is used for the synthesis of DNA. So during DNA replication, the polymerase enzyme is basically required because there's going to be a synthesis of DNA in this procedure. We have ligases that joins two DNA strands at the end to form the phosphodiester bonds. The next tool for recombinant DNA technology is what we call the vectors. And as the name implies, the vectors are basically molecules that carries our gene of interest into another organism. So a vector like plasmid, which is the extra chromosomal pattern or let's say molecule found in bacteria, can serve as a vector because we can insert our gene of interest inside plasmid and we can incorporate that plasmid into a bacteria so that there can be further replication to produce our gene of interest, a lot of copies. Known one is the bacteriophages, which sometimes also act as vectors. But the most used vectors in molecular biology is the plasmids. Now, for a good vector to be used, it must have what you call an origin of replication. That is a site where there can be division of the cell because when it's being inserted into a host organism, it will undergo replication to produce several copies of the same plasmid. So a good vector must have an origin of replication. It must have what we call a selectable marker. Basically, this section is basically an antibiotic resistant gene that is basically used to isolate the vectors that picked our gene of interest from, or let's say the bacteria that picked our plasmid, or the bacteria that picked our the vector from that didn't pick our vector. You will go to the procedure for recombinant DNA technology and you will understand this better. The next site is what we call the registration enzyme site. Now, a good vector must have a site where the registration enzyme can cut so we can insert our gene of interest inside there. The last one is what we call the host organism. The last two is the host organism. Now, the host organism is that which can be incorporated into the vector or which the vector can be incorporated into that it can be used to synthesize a lot of copies of our vector. So we can have several copies of the host organism containing our gene of interest, also containing what our vector. So the known or the most used host organism in recombinant DNA technology are bacteria, because bacteria have an extra chromosomal molecule called for the plasmid, and this plasmid can be incorporated into a vector, what do you call it, our gene of interest, so that this one can find itself in the bacteria that it can replicate, or let's say it can divide threshold so that it can have several copies of our gene of interest inside the bacteria. So the host organism we say is an organism that can host our vector for it to undergo several or further replication. Now let's look at the procedures involved in recombinant DNA technology. After this, we are going to have a pictorial view of how the procedure is done. The first step of you trying to incorporate a gene of interest into a vector and further into a bacteria is isolation. The procedure is very simple. You have to get your gene of interest, you incorporate it into the plasmid, or let's say the vector, and you incorporate that too into the bacteria or the host. So the first procedure is what we call the isolation of genetic material. Let's say if you have a particular gene of interest you want to make copies of in a DNA or in a bacteria or plasmid. The first thing is to isolate the genetic material, which is the DNA. So here we mean the DNA strand of interest. So the genetic material as shown here is the DNA strand of interest. So from that, so let's say is it from a human cell, an animal cell, or plant, you have to isolate that gene of interest without any impurities. That is the pure form of that gene of interest. The pure form of that DNA, either from plant or animals. You have to get the pure form of that, and that's what we call the isolation. The second step is what we call the cutting of the gene at the recognition site. So here is more like two steps, same time. You are going to cut the section on the plasmid that can incorporate or that can swallow the gene of interest. And the cutting is what we use the restriction enzyme. The restriction enzyme does the cutting so that it can incorporate or accept in there our gene of interest. The cutting is basically done by the restriction enzymes, which we call the molecular scissors. The next step is the amplification of the gene of interest. So let's say if you have one gene of interest from our um, our source and you want to amplify it, you have to get a lot of copies of that, not that much, but you want to get enough copies of our gene of interest isolated so you can input into a known or good amount of bacteria, or let's say a host, or let's say even a plasmid, so that you can get a lot of that. The amplification is done using the PCR. In this PCR, we are yet to do a video on how PCR machine works. So don't forget to subscribe so that when you upload this video, you will get notification of that. It's a polymerase chain reaction mechanism. So the name is polymerase. That uses the polymerase enzyme. So here the polymerase enzyme that mentioned up here comes here. So it's a polymerase chain reaction that is used as a machine which is used to amplify a particular gene of interest or DNA, more like an in vitro without in an organism. It involves series of steps and mechanisms which we are going to discuss and do a video on in the next video so that you can get and appreciate better what we mean here. So this one is the first. The next one is the amplification of your gene of interest using the PCR or the polymerase chain reaction machine. Finally, it is more of a ligation procedure. So the next one is the ligation. With this ligation, you more incorporate the gene of interest into the plasmid or the vector. So when you plasmid or you send or you put the gene of interest into the plasmid or the vector, then you ligate it using the ligase enzyme. We're going to look at that in the next slide. The last one is the insertion of our plasmid or the vector into the host organism. So let's have a pictorial view of how the method works. It's very simple. Here we have the bacteria plasmid. We have here the ampicillin resistant gene. So here this section of the plasmid is what we call the selectable marker. This section is resistant to ampicillin. That means that if you're able to get a lot of bacteria picking up this plasmid, when we get a lot of that bacteria, some might have the gene of interest, some might not have the plasmid or the vector of interest. If you add an ampicillin to that mixture or that combination, we are going to have those without this plasmid dying because they don't have a gene that can resist ampicillin. The ampicillin is an antibiotic that can kill a bacteria. So if bacteria lacks this selectable marker, or let's say it lacks this plasmid, with this selectable marker, it will die automatically. The next one is the LAX-Z gene. So here basically is the restriction site. of a registration enzyme where it can be cut and can incorporate the gene of interest into. So here, as shown here, this section with the LAX-Z gene is more of a registration enzyme site which can be cut. This type of cutting as shown here is called the sticky end cutting. So here the sticky end, the sticky end is on both the plasmid and the gene of interest. So we have our gene coming from a human being. We also cut the same gene with the registration enzyme. So we get a sticky end and a sticky end. Now from here to here, we can then send the gene of interest into the bacterial plasmid. So we get this. So now the resulting here is what we call the recombinant DNA. This one is a DNA, but it's a recombinant one because it is a recombined DNA DNA that is from different sources. So this one we have here the plasmid. This side is the selectable marker. Here is the newly cloned gene. So here is more like a cloning mechanism. So when you have this, then maybe you add the ligase so that you can ligate the bond between the two DNA strands. The next step is what we call the incorporation or transformation. So we can then transform or transfer this what do you call it, the plasmid into a bacteria. A lot of mechanisms are used for that. We can have micro injection, we can have gun shooting, we can have the use of calcium ions. So we more of use this, insert this plasmid into the bacteria by several mechanisms. So here, this is our bacteria, and we assume this our bacteria has our gene of interest or has our plasmid. This is the plasmid with our gene of interest, and this bacteria has been incorporated into or incorporated with the gene of interest or the plasmid. If you have a lot of these bacteria, some of them might pick this plasmid and some might not pick this plasmid. So if you have this bacteria and we grow this bacteria in a media, they will all grow. So let's say in this case, these ones are the ones without the plasmid. How can we then separate those without the plasmid from those with the plasmid? That is where the role of the selectable marker comes in. If you have this mixture and you add an ampicillin, since the plasmid has an ampicillin resistant gene, those that pick the bacteria or the plasmid will not die, but those without the plasmid will die because they don't have any gene to resist ampicillin antibiotic. So basically that's how DNA technology works. It's very simple. So after going in a separation with the ampicillin, you can then isolate and purify this bacteria. So if this bacteria has our gene of interest, let's say that of the gene of, let's say, an insulin, this bacteria can automatically synthesize insulin in its molecule or in its system or in its cell, so it can be isolated and used for, let's say, a human being that lacks or that needs that insulin to cure his or her diabetes. So it's very simple. That's how basically recombinant DNA technology works. Let's look at another pictorial view of the method. So now we have here our transformed bacteria with our gene of interest. So this is the gene of interest with the bacteria. Let's say this gene of interest we sent in here was that of a plant. Now this bacteria can modify this gene to form a DNA molecule, or sorry, a chromosome, because this one is a DNA molecule, and DNA molecule forms a chromosome in bacteria. And this one can be isolated if it is a seed in this case and planted. So that's how genetic modified foods are synthesized. So this one is a typical genetic engineering mechanism where genetic modified food is synthesized. If you have a particular gene, or let's say a seed that can grow better and resist much harsher environmental conditions, we can use this method to synthesize a lot of genes of that seed so we can have it in a healthy form. Let's look at the pictorial form of the method. Very simple, very summarized. So this is what we looked at earlier. It's just very simple. We have a human cell and this is our gene of interest from a human cell. Then we cut the human cell to provide a sticky end gene of interest. Then we take a plasmid from bacteria. Then we cut the bacteria with the equal R1 registration enzyme to produce also a sticky end. We can then incorporate this um, DNA of interest or gene of interest from human cell into the plasmid to produce a recombinant DNA. Now, this is the recombinant DNA. The, this part is it the green part, so or the blue part rather comes from the bacteria, and this part comes from the human being. So this is why we say it's a recombinant DNA because it has a recombined DNA from bacteria and human source. We can then incorporate this plasmid into the bacteria, and this bacteria can undergo several divisions when cultured to produce millions of the same bacteria believing to contain our gene of interest. So this mechanism can be used for an in vivo DNA replication, and the in vitro one is that of the polymerase chain reaction, which I'm going to look at in the next video. And also this same bacteria can use this gene to synthesize our protein of interest, what is, let's see, so that we can isolate that protein from this bacteria and use it for the cure of disease. Thanks for watching. This is just a summary on polymerase, or sorry, recombinant DNA technology. We are going to do the next video on polymerase chain reaction, the mechanism involved, and how it can be aided in gene cloning and genetic engineering. Don't forget to subscribe, like, and press the notification bell after watching so you can receive every bit of update from the channel. Enox tutorials. Thank you.